Hello, how are you? Today, inshallah, we will complete with 15.3 application of genetic engineering part 3. In the last lectures, we talked about the genetic modified plants, genetic modified animals. We talked about the benefits of recombinant DNA in health. And today, inshallah, we will complete with genetic testings and DNA fingerprinting. So let's complete. For today's lectures, my objective is summarize the process of the DNA fingerprinting and explain its uses. Okay, so in the beginning we need to use the genetic testing and to study the genetic testing procedure. What do you mean by genetic testing? Genetic testing can be used to determine if two prospective parents are carrying the alleles for a genetic disorder like cystic fibrosis. As you remember, cystic fibrosis, a disease that is showed after a mutation, a deletion with an amino acids, which is 3 base pair deletion. And the cystic fibrosis allele has slightly different DNA sequence from its normal counterpart. Genetic tests use labeled DNA probes that can detect and distinguish the complementary base sequences found in the disease-causing allele. So we need to use the labeled DNA probe. The labeled DNA probe here is going to be in 2 colors, okay? Some genetic testing search for changes in cutting sites of restriction enzyme, while others use PCR to detect differences between the length of normal and abnormal alleles. So we need to use either cutting site of the restriction enzyme or to detect the differences between the length of the normal and abnormal allele. At the same time, genetic tests are now available for diagnosing hundreds of disorders. Can you mention 1 or 2 or even 3 disorders that can be screened by the genetic test? Can you? Can you search for some of the disorders that can be caused by genetics? So some of the disorders that can be discovered by genetic tests? I want you to search and tell me with the answer, okay? Let's begin with the examining active gene. The same gene are not active in every cell. We have to know that the same gene is not active in every cell. And by studying which genes are active and which genes are inactive in different cells, scientists can understand how the cells function normally and what happens when genes don't work as they should. We need to know where exactly the gene is where exactly in which cell the gene is active and in which cell the gene is inactive. Scientists also use the DNA microarray technology to study hundreds or even thousands of genes at once to understand their activity levels. So the technology that is used here is DNA microarray. Make sure that you understand that. DNA microarray technology to study hundreds or even thousands of genes at once, okay? Let's check how the DNA microarray is done. DNA microarray is a glass slide or silicon chip. So glass slide or silicon chip to which spots of single-stranded DNA have been tightly attached. So on the chip, single-strand DNA will be attached. Then spots contain a different DNA fragments and differently colored. Different fragments with differently colored tags are used to label the source of the DNA. So each DNA source, each strand will be labeled with a different color according to the source. For example, to compare the genes in cancer cells with genes of a normal cell, the mRNA would first be isolated. So the first step to isolate the mRNA from both types. So this is the mRNA that will be isolated from the cancerous cell, and this is the mRNA that will be isolated from the normal cells. Then we will use enzymes to prepare complementary DNA molecule. So in the beginning we have the mRNA, and then we need to form the cDNA, which is called complementary DNA, by the using of specific enzymes. So the cDNA here for the cancerous cells is in the red color, and the cDNA here for the normal cells in the green color. And you have to know that the red color and the green color here is labeling, radio labeling, or tagging, okay? So again, the cancerous cells are labeled by the red color, and the normal cells are labeled by the green color. Then both of the samples of the labeled DNA would be mixed together. So we need to mix both of the DNA samples, and they would be then complete to bind to the complementary DNA sequence already in the microarray. So they need to bind with the complementary DNA that is found already in the microarray. Okay? So once the DNA or the cDNA is bounded already with the complementary DNA that is found already in the microarray, it will form double DNA strands. As you can see here, this is the cancerous cells, so the cancerous cDNA, and this is the normal cDNA when it's bound to the complementary sequence on the plate. After that, we need to make a film, okay? If the cancer cells produce more of a particular form of the mRNA, then more red labeled molecule will bind at the spot for the gene, which is going to turn into red, okay? This is a screening for the microarray plates, where the normal cell produce more mRNA for another gene. The spot will be green. So the spot here, green for the normal, and the spot here, red for the cancerous cells, where there is no difference between the two cell types. The spot will be yellow because it contains both colors. So the yellow here contain both colors, the red and the green in the same time. The other thing that we can use the DNA fingerprinting by it, which is the personal identification. Personal identification, I can say this person related to this parent, okay? So how is DNA used to identify individuals? How DNA is used to identify individuals? The answer is DNA fingerprinting analyzes section of DNA that may have little or no function, but that vary widely from one different to another. So, as you know, the DNA composed of coded segments and non-coded segments. The coded segments are coded for protein, and non-coded segments are not coded for protein. And the DNA fingerprinting, we are checking about the non-coded part of the DNA, non-coded part. So what we will check here, we will check the repetition of the non-coded gene. Okay? So this is the DNA. This is the DNA sequence. And here in the non-coding sequence, we can see repeats of a specific sequence. And that repeats here in this person is different from the other person. As you can see here, we can see 12 repeats here and 9 repeats here in the same DNA sequence. This is how we can compare between two suspects or between two samples. Chromosomes contain many regions with repeated DNA sequence that do not code for a protein. So you have to know that repeated sequence is not coded for proteins. These vary from person to person. In the first sample, as you can see here, 12 and here 9. In the other sequence, 4 repeats and 6 repeats in the other part. So DNA fingerprinting can be used to identify individuals by analyzing these sections of DNA that may have little or no function, but that vary widely from one individual to another. So there is no function from these sequences, no function from this repetition, but it's vary from person 1 to 2 to 3 to between all of the community or the population. And we can use this technology to compare between the suspects in the criminal cases. In DNA fingerprinting, restriction enzyme first cut a small sample, the human DNA, into fragments containing genes and repeats. Note that the repeated fragments from these two samples are different of length. So we have to know that the repeated sequence have a different length, okay? Then we will use a gel electrophoresis. For the first step, we have to know that we need to use specific enzyme that will cut the DNA into fragment, which is called restriction enzyme here. And once this is the first step, miss, then we will use the gel electrophoresis, okay? Once we use the gel electrophoresis, you have to know that you have to apply the DNA here on the negative side, and the DNA will migrate toward the positive side. This is the negative and this is the positive, by the way, okay? So the direction of migration, the DNA will migrate to this side. Why the DNA migrate from the negative to the positive? Do you know? The DNA migrate from the negative to the positive because the DNA contain negative charge, okay? The DNA contain negative charge. And once the DNA is a negatively charged, it will migrate from the negative to the positive because of the pulling reaction between the positive and the negative. And once the electrophoresis is finished, we need to read this gel by X-ray. We will make an X-ray diffraction, and once the X-ray is done, we will read the picture. Here, this is sample 1, this is sample 2, and this is the evidence sample. We need to check this evidence sample for whom? Is it for suspected 1 or sample 1 or 2? As you can see here, this is a band, another band, also here 2 bands, and here also 2 bands. Which of the 2 samples have the same bands at the same length or the same size? Is it sample number 2 or sample number 1? Can we see or can we check? And so by this technique, we can say that sample E related to sample 2, okay? You have to know how we can read the gel electrophoresis, okay? You have to know how we can say this sample is related to suspected 1 or 2, okay? Or sample 1 or 2. Again, you have to know that a DNA probe then detected the fragment that have highly variable region revealing a series of variously sized DNA bands. So this is bands, this is a band, okay? And this is a band here. And you can say this band similar to S2, this band similar to S2, and so on. So this technology is really used in the forensic sciences. You have to know that this technique is really precise and reliable technique on the forensic sciences because they will use it to identify the criminal in the crime scene because there is evidences in the crime scene and they will use the evidences to compare between the suspected 1 or 2 or even a 3. DNA fingerprinting has helped solve the crimes, convict criminals, and even overturn wrongful conviction. And by the way, it's helped in more than 110 wrongfully convicted prisoners from death sentences. Let's find how we can establish a relationship. When genes are passed from parent to child, genetic recombination scrambles the molecular markers used for DNA fingerprinting. So ancestry can be difficult to trace. So your DNA is similar to your father and mother DNA together. So your DNA must be 50 from your father and 50 from your mother. And you have to know that, okay? While the DNA is passed from your parents to you, there is small change will happen to your DNA and that will not affect your health or your genes, okay? The Y chromosomes that must be in the males never undergoes a crossing over and only males carry it. So there is no crossing over in the Y chromosome like other chromosomes like chromosome 1, 2, 3, 4 and even 5 till pair 21, 22. But in the pair 23, the Y chromosome will not undergo crossing over. And you have to know that Y chromosomes pass directly from father to son with a few changes, very few changes, okay? Another thing that can pass from the mother to the child directly without any help from the father is called the mitochondrial DNA. The mitochondrial DNA is passed from the mother to the child because it's already found in the cell or the egg cell of the mother, in the cytoplasm of the egg cell of the mother, and it's not found in the sperm, okay? So it's found just in the mother DNA. So in this case, it will be passed to the offspring. So your mitochondrial DNA is similar to your mother, not your father, okay? And your brothers or sisters' DNA, mitochondrial DNA, is similar to your mother and in the same time. So mitochondrial DNA is passed directly from your mother, from the mother to the child. So your mitochondrial DNA is same as your mother, not your father. Okay, so by this point we can say that the relationships between you and your parent is wide. Your DNA, your mitochondrial DNA is similar to your mother. If you are a male, your Y chromosome is from your father without or with a few or very few small changes, and your whole DNA, nuclear DNA, is composed from your mother and your father DNA. At this point, we complete the applications of genetic engineering. Hopefully you will go back to the part 1 and part 2 and connect all of the rules together. Okay, have a nice day and thank you.